Today, we're doing a deep dive into cancer research, and we're tackling one of the biggest challenges in precision medicine.
Right. It's really about connecting the dots.
Exactly.
Mm.
Connecting what's broken in a cancer cell's DNA to how that actually, drives the disease. We're talking about signaling pathways.
The cell's wiring, basically. The commands that tell a cell to grow, to survive, to spread. It's the functional core of cancer biology.
And we have just incredible amounts of genomic data now, thanks to projects like the Cancer Genome Atlas.
Oh, absolutely. We know about specific mutations. We know about something called copy number alterations, or CNAs.
That's where a cell has too many or too few copies of a certain gene.
Exactly. But here's the problem. Knowing the genotype, the DNA code, doesn't always tell you the phenotype. It doesn't tell you what the cell is functionally doing.
Okay, let's unpack this. We have this mountain of data, but trying to use it to predict, say, patient outcome or drug response has been, well, a bit frustrating.
It has. Because for thousands of samples, we can't do direct measurements of activity. It's just not practical.
So we're forced to guess, to infer it from gene expression data.
Precisely. And the standard methods we've been using to do that have some pretty serious structural flaws.
How so? What are we talking about?
Well, if you use a traditional pathway analysis tool, you're usually just mapping the expression level of the genes that make up the signaling pathway itself.
So if a gene for a pathway protein is highly expressed, you just assume the pathway is active.
You assume it's active. And that sounds reasonable on the surface, but that's where the trouble really starts.
I had a feeling you were going to say that.
It's a twofold problem. First, this whole approach ignores something called post-translational modifications. PTMs.
Right. So the protein is there, but it's not necessarily switched on.
Exactly. A protein can be highly present in the cell high expression, but if it hasn't been chemically activated, usually by adding a phosphate group, it's just dormant.
So it's like we're looking at the detailed schematic for the house's wiring, but we have no idea if the main power is actually on.
That's the perfect analogy. You see the light switch on the wall, but you don't know if current is flowing through it. The second flaw is what we call context specificity.
Meaning a finding from one experiment doesn't apply to another.
You've got it. A gene signature you find in one specific cell line, treated for 48 hours in a lab, might be totally useless when you try to apply it to a real patient's tumor. We need something more general.
Which brings us to the methodology we're diving into today. It's a new computational approach called PROGENy.
PROGENy. It stands for Pathway Responsive Genes. And it's a really elegant conceptual shift.
How so?
Instead of worrying about whether the light switch is present, PROGENy looks downstream. It focuses on the genes that consistently respond whenever that pathway gets switched on.
So it measures the light coming on, not the switch itself. It's a functional readout.
The authors call it a signaling footprint. It's the echo of the activity.
Okay, that makes a ton of sense. But how do you find a footprint that's general enough to overcome that context specificity problem you just mentioned?
That's the real innovation here. To find that general footprint, they embraced heterogeneity. They didn't just use one or two experiments.
I used a lot more.
They built a massive compendium. They took 208 public data submissions, which covered 568 different perturbation experiments, all on microarrays. At the time, it was the largest study of its kind.
Wow. 568 experiments. And what were these experiments doing?
They were all designed to prod a specific signaling pathway. The researchers focused on 11 really critical cancer-relevant pathways, things like EGFR, MAPK, PI3K, the P53 response.
And the diversity of the experiments was the key.
It was the filter. By looking across hundreds of experiments where, say, the MAPK pathway was activated in this cell line and that tissue under these conditions, they could ask a simple question.
What genes always change?
What genes always change their expression, regardless of the cell type, the timing, or how you activated the pathway? They were looking for the universal truth.
And they found it.
They did. They used a statistical model to distill all that data down to the top 100 responsive genes for each of those 11 pathways. This is the common core signature.
So these 100 genes are the most stable, most consistent reflection of that pathway being active.
Yes. And here is the most crucial detail, the part that validates the entire idea.
Okay.
These 100 responsive genes have almost no overlap with the genes that actually encode the signaling proteins themselves.
Wait, really? So the response signature is completely different from the pathway's own components?
Almost completely. It proves you can't just substitute one for the other. They are two distinct biological processes. Activation and expression should be treated separately.
So how do they prove this footprint actually measures real activity?
They used an orthogonal method, the gold standard, phosphoproteomics.
Yeah.
They took a cell line, activated a pathway, and then directly measured the phosphorylation of the proteins.
They measured the actual power surge at the breaker.
They did. And then they asked, does our PROGENy score match that surge?
And did it.
It did, perfectly. When they induced MPK signaling, the MAPK PROGENy score shot up, and that lined up exactly with the measured phosphorylation of key proteins like MEK and ARK. It gave them huge confidence that this isn't just transcriptional noise, it's true functional activation.
Here's where it gets really interesting. Moving from a controlled lab setting to messy, large-scale patient data. Let's talk about the Cancer Genome Atlas, TCGA. How did PROGENy do there?
It performed exceptionally well. The logic is simple. If the method works, patients who have a known driver mutation that activates a pathway should have a higher PROGENy score for that pathway.
And that's what you see.
That's what they saw. For instance, EGFR amplifications, a common event in some cancers, led to high scores for both the EGFR pathway and the MEPK pathway.
Which makes sense since that's the downstream cascade.
Exactly. Or you look at KRAS and BRAF mutations, those notorious drivers of cell growth.
High MBK activity.
High inferred MPK and EGFR activity. It just works.
But the really cool insights must come from the more complex genes, like TP53, the famous tumor suppressor.
Absolutely. When TP53 is mutated, you see exactly what you'd expect. The PROGENy score for the P53 DNA damage response pathway plummets.
The cell loses its ability to respond to damage.
Right, but it also captured a critical secondary effect. They saw a simultaneous activation of the PI3K and hypoxia pathways.
So it's not just that something is lost, something new is gained.
Exactly. It's functional evidence for the idea that many TP53 mutations are not just loss of function, they are gain of function. They actively turn on new cancer-promoting pathways. It's a dual insight you'd miss with other methods.
That's fascinating. And what about VHL, the von Hippel-Lindau gene?
This might be the single strongest case for PROGENy superiority. VHL mutations, which you see a lot in kidney cancer, cause a protein called HIF1 alpha to become stable.
Which tricks the cell into thinking it's in a low oxygen or hypoxic state.
Right, and PROGENy found that VHL mutations were uniquely and powerfully associated with the induction of hypoxic genes.
Okay, why is that so important?
Because the mechanism is protein stabilization, not a change in gene expression of VHL itself. HIF1 alpha just stops being degraded. Traditional methods that just look at RNA levels really struggle to find this link.
But PROGENy sees it clear as day.
It sees the downstream output, it sees the footprint of hypoxia. It's capturing a biological mechanism that is otherwise invisible to standard RNA analysis.
Okay, so that's advantage 1, accurately mapping the function of driver mutations. Let's move to advantage 2. Predicting drug sensitivity.
Right, using the GDSC data, the genomics of drug sensitivity in cancer, this is where you test hundreds of cell lines against hundreds of drugs.
And PROGENy's scores predicted drug response better.
Significantly better. And the strongest hit they found perfectly illustrates the power of measuring activity, not just presence.
Let's hear it.
It was the link between the MDM2 inhibitor, Nutlin-3A, and the P53 responsive genes.
So Nutlin-3A is a drug that works by reactivating P53.
Precisely. So it only works if the cell has a functional active P53 pathway to begin with. And PROGENy captured that functional dependency perfectly, while just looking at the expression of the component genes, missed it entirely.
This leads to what might be the biggest clinical idea in the whole study. It's about refining biomarkers.
This is the paradigm shift. Take the MEK inhibitor, trametinib. It's a targeted therapy.
Right, usually given to patients with BRAF or KRAS mutations.
Correct. But what PROGENy showed was incredible. Cell lines that had high active MEK scores, even if they did not have the classic BRAF or KRAS mutation,
They still responded to the drug.
they were 15 times more sensitive to trametinib than cell lines that did have the mutation, but according to PROGENy, had an inactive MEK pathway.
Hold on. That is a huge finding. You're saying the functional score was a much better predictor of drug response than the genetic mutation itself.
That is precisely the implication. The presence of a mutation is irrelevant if that pathway isn't functionally driving the cell. PROGENy lets us move from a static genetic prediction to a dynamic activity-based one.
Which means we could be giving these drugs to the right people and stop giving them to people who won't benefit even if they have the right mutation on paper.
That's the goal of precision medicine.
Okay, finally, let's talk about the ultimate endpoint, patient survival. How did the scores line up with TCGA survival data?
This is where it all comes together. PROGENy was able to clearly separate the pathways that drive bad outcomes from those that don't.
So the oncogenic driver pathways.
Things like EGFR, MAPK, PI3K, and hypoxia. High activity in any of those was significantly associated with decreased patient survival.
And it could distinguish those from, say, a cell death pathway.
Yes.
Right.
It showed that activity in the trail apoptosis pathway, which is a pro-death mechanism, was associated with increased survival. A clean functional separation that other methods really struggle to make consistently.
Wow. So high activity in growth pathways means worse survival. High activity in death pathways means better survival.
It's biologically intuitive, and PROGENy quantified it. We saw this in specific cancers, too. In adrenocortical carcinoma, or ACC, higher p53 activity was strongly linked to better survival.
Which makes sense for that tumor type.
Right, because those tumors often don't have the aggressive gain-of-function variants. So this allows us to stratify patients. In some cases, there was over a 25% difference in one-year survival between patients in the top and bottom quartile of a pathway score.
So what does this all mean if we boil it down for everyone listening?
It means that PROGENy, this tool built from a consensus of hundreds of messy, heterogeneous experiments, gives us a robust, general, and clear measure of pathway activity.
And the big takeaway seems to be, if you want to infer pathway activity, predict drug response, or stratify patients,
You should be measuring the downstream gene response signature, the footprint, not just the expression level of the upstream signaling molecules. The functional footprint is what matters.
it fills in that critical gap between the genetic code and what the cell is actually doing.
Yes, exactly.
Now, you mentioned that PROGENy's strength is its generality. But the source material pointed out a potential trade-off. This seems like a great final thought for our listeners.
Correct. The authors built this incredibly robust tool by finding the common core response across many, many conditions. But they did note that if you have a very specific question
About one specific tissue, for example.
right, a signature derived only from experiments in that one tissue might capture certain nuances better in that specific context.
And that raises a really important question for the future, for clinical use.
It does.
If you're a clinician treating, say, lung adenocarcinoma, when do you rely on a general, robust signature like PROGENy? And when do you turn to a highly specialized module that might be more accurate for that one disease, but completely useless everywhere else?
What's the trade-off between generality and specificity? Balancing that for individual patients, that's the next great challenge for precision oncology.
A fantastic, detailed deep dive. Thank you for walking us through how we can get a much clearer picture of what's happening inside a cancer cell.
My pleasure. It was fun.
And thank you for joining us for this in deep dive. We'll be back soon with more critical information distilled for you.